Thank you for inviting me to speak today. I'm Greg. I'm a postdoc at the University of Liverpool, and my research focuses on exploring the spread of antimicrobial resistance genes in the hospital sink drain microbiome. So when I talk about the hospital sink drain microbiome, I primarily mean the community that lives inside sink traps. So underneath sinks, you'll have seen in the pipework, you either get these P traps or bottle traps, and they're basically just reservoirs of water that prevent odour coming up from the sewer. When gases come up from the sewer, it stops it coming up through the drain. But as you can imagine, once you install these traps, this standing water quickly starts growing lots of bacteria, and you get these really thick biofilms starting to grow inside these traps. And in a hospital, you can imagine that's a lot of bacteria from the hospital environment. A lot of opportunistic pathogens start forming these biofilms. So these traps are a huge reservoir of opportunistic pathogens in hospitals. And we also know that biofilms are a prime location for plasmid transfer because of the tight cell-to-cell contact that you can get lots of horizontal gene transfer, particularly conjugation of plasmids. And these can often carry antimicrobial resistance genes. So not only could these traps be an important reservoir of opportunistic pathogens, but they could also be a reservoir where there's a lot of plasmid transfer happening and a lot of spread of resistance genes. And just zooming out, thinking about what goes down a hospital sink, you've got constant microbes going down from the hospital environment. You've got nutrients going down. There's lots of chemicals going down, and that could be disinfectants that are regularly used, or even drugs in a hospital. And you can imagine that can cause a big selection pressure on the biofilm that could increase the transfer of resistance genes. And every time you run the tap, you're getting splashes and aerosols that can cause infection in immunocompromised patients nearby. And any infection in the hospital can end up going back into the sink, and outbreaks can be prolonged by bacteria living in these sinks and being hard to get rid of in the hospital environment. And also all the sinks are connected with the plumbing network, and we know that bacteria can move along the drains, so we can get adjacent sinks nearby starting to grow the same bacteria. So there's obviously lots of interventions in place to try and prevent the risk of causing infection from these sinks. Often you'll see in hospitals that this sink is for hand washing only, and that's to try and prevent nutrients going down to allow the biofilm to grow better. But actually shortly after I started this postdoc, I ended up in hospital for a couple of months. So I started noticing things from the sinks nearby, and I started taking some notes. That I've called Greg's Hospital Sink Undercover Observations, and I'll just show you some of the highlights. So you can get visitors pouring apple juice and coffee down the sink in the ICU. So whilst clinicians and clinical staff will be following these signs that it's for hand washing only, patients and visitors will see a sink as a sink and use it as such. And by visitors, I primarily mean my mum then. I often saw the cleaners wiping the sinks with a cloth and then using the same cloth to wipe the rest of the room, including door handles. So you can see how bacteria in the sink could then be transferred to the rest of the hospital environment. At one point, a cleaner was mopping the floor outside my side room, and then came into the side room and rinsed the mop down the sink in my room. So that then all the microbes from the ward was now in this patient's sink. At one point, I washed up a bowl of spaghetti bolognese in my hand washing sink because I had nowhere else to do it. So you can see the nutrients going down, and patients will be doing that. And the worst one was at one point I had a vancomycin drip and it started leaking everywhere, and the nurse caught a load of it in a cup and then poured it down the sink. So that was 10 ml of pure vancomycin going straight down the sink, and you can imagine the selection pressure that's putting onto the biofilm. And I'm not showing this to complain about anything. I'm just showing that in the real world, there are these interventions in place, but these things happen, and the sinks are exposed to these drugs and nutrients all the time. And we know from studies that there are lots of opportunistic pathogens originating in sinks that do cause infections. So there's studies that show that this infection was caused by a bacteria that was found in the sink just before, or lots of studies showing outbreaks lasting months or years even, and they don't end until all the sinks on the ward are replaced. So it shows that the sinks have a really important role in acting as a reservoir for outbreak strains during prolonged outbreaks. But often these studies are observational or just focused on the epidemiology. And our study, we really want to focus on the biology of what's going on in these sinks and find out what's driving antimicrobial resistance in this environment. So for our study, we've been collecting lots of sink trap samples from Liverpool hospitals. This is just a nice photo I like to show of just some of the samples we've collected, just to show the diversity of colours you get from this trap water. You never know what it's going to look like when you start sucking it up. Some of it's quite disgusting. But we've characterised all of these with metagenomic sequencing, and we've also isolated hundreds of strains from these. And we're now using these samples and strains that we've got in experimental models to try and see what the main drivers of antimicrobial resistance are in these microbiomes. So just quickly, I'll show one of the experimental models I've been doing this year. So it's this real world model, and it's basically down in UKHSA in Porton Down, they have Building 44, which is this fake hospital. And it's basically a full hospital ward. It's got every room that you'd imagine on a hospital ward, and it's all built to NHS spec. But there's no patients there other than these dummies, so you can't infect anyone. So you're allowed to run experiments there, and we can use this as a sort of mock hospital setting with real sinks in there. We wanted to see whether the use of disinfectant over time could increase horizontal gene transfer. So this is a floor plan of Building 44, and you can see there's lots of sinks, and we used half of these sinks as control sinks that weren't treated with any disinfectant, and half that received hospital grade disinfectant every week. And all these sinks got sort of the taps flushed and a little bit of nutrients going down each day to simulate a real world conditions. But they all started with the same starting traps. We inoculated the traps with brand new traps with pooled samples from real hospital sinks that we've got to sort of grow a biofilm for a couple of months before installing them all. So just some very preliminary data that I've been working on in the last couple of weeks. Basically, I sampled those every week, and we've done some metagenomics on these longitudinal samples. And just in terms of diversity, we can see that the disinfectant did have an effect on the community in these sinks. So looking at the alpha diversity, the control sinks in pink sort of stayed quite stable throughout, but the disinfectant sinks did decrease in alpha diversity over time, and that's both in species richness and in the Shannon and Simpson index. And then looking at some beta diversity, this plot shows each point here is a weighted unifrac distance. And we can basically see that the treatment with disinfectant did cause the samples to diverge over time. So in axis 1 here, we can see that the points are separated by treatment. And in axis 2, it's sort of separating it by time. So we've got the early weeks at the top and the later weeks at the bottom. So the samples, clearly, the community composition did change over time, but clearly the treatment with disinfectant did have an impact on the communities there. And just looking at how it impacted them, very quickly for this short talk, I just show this taxonomic profiles over time with the control sinks at the top and the disinfectants at the bottom. And each colour here represents a different genus in the sinks. And you don't need to see all of it because we haven't got too much time, but just to highlight some of them, we can see that Aeromonas in this turquoise colour was quite abundant in all of the sinks at the beginning, but in the control sinks it decreased in abundance over time as the other genera increased. But in the disinfectant sinks we didn't see that decrease. It actually remained the same relative abundance or increased. And this is an opportunistic pathogen that's often found in hospital sinks. And then in yellow, we've got Aquapseudomonas, which is very closely related to Pseudomonas, which is an opportunistic pathogen that is often found in sinks and is known to cause infection from hospital sinks. And we see that in the disinfectant-treated sinks, the Aquapseudomonas greatly increased during the experiment, but we didn't see that in the control. So we can see that the communities definitely differ when treated with disinfectant, and it seems that opportunistic pathogen species are increasing in abundance compared to without disinfectant. And then very quickly, just looking at the abundance of resistance genes, we can see that with disinfectant treatment, the antimicrobial resistance genes were increased compared to control, and the plasmid genes and biocide resistance and heavy metal resistance genes were greatly increased compared to the control sinks. So the disinfectant use clearly increases opportunistic pathogen abundance in this community, and it also increases a relative abundance of resistance genes. So the next stuff I'm going to be doing is looking at plasmid transfer over time, but I haven't got that data for you today, unfortunately. But that's what I'm working on now. So thank you very much for listening, and just thank you to my supervisor, Jamie, and John and Danielle at Liverpool Hospitals that have helped us sample all the sinks. And Jenny Moore runs Building 44 down in UKHSA. So thank you to her. So thank you very much, and I'm happy to take any questions later on.